Hi, this is Kevin Hrusovsky. I want to welcome everybody to our webinar to talk about the SPX. We're really excited about this next generation platform that we believe has great multiplexing capabilities that also gives the same level of sensitivity as previous Simoa technology. So we're pretty excited about the evolution. So today is intended to just give you somewhat of an online visibility into it. And Mark Roskey will be doing most of the heavy lifting today to describe the SPX and how far we've come in the last, I'd say, 6 to 9 months. But I will lead it in with just some updates because as a company, we are making a lot of advances on many different fronts. And I want to just provide a little bit of a perspective around some of that. Starting first, that as you know, we have got this secondary kind of organization called Powering Precision Health. It's independent of Quanterix, but our next major symposium is going to be in Barcelona on November 19th and 20th. And we just really want to make sure everyone's aware that we started this really around trying to transform invasive late-stage detection of cancer and neurodegeneration into what we would consider to be much earlier detection of disease cascades, but doing it semi-non-invasively via blood samples, saliva, and others. Our focus based on this next slide is just to say that our primary focus is cancer and neurology. We feel like those are the two diseases that are the latest stage in detecting them. And by the time you detect them, it's typically very difficult for drugs to have any benefit because it's so late stage. And for that reason, we're deploying a lot of our sensitivity, both in invasiveness and in early detection. We're trying to hone in really on cancer and neurology. And what's most important around the SPX launch is it's better suited for cancer research because we can multiplex at a higher number. And we've spent a lot of time optimizing and validating our cytokines, most of them at baseline levels, which is pretty exciting. This slide just illustrates that as a company 4.5 years ago, we decided to redirect it away from pure diagnostics and then move into what we call the research markets, helping pharma, biotech, and universities and academic centers begin to understand the technology and create peer-reviewed publications. We started in neurology, and with the launch of the SPX, we're now trying to evolve rapidly into oncology, particularly around immunotherapies, where we feel there's an incredible opportunity to improve response levels from maybe at a 10% level and toxicity levels of cytokine release that could also be at the same level, 10%, to increase, obviously the effectiveness, efficacy, but also to significantly reduce and seeing the toxicity much earlier when you can save patients. And so this next slide just summarizes the product lines that we have today. And you know that the bead technology, which has been primarily deployed up to 4-plex and maybe we have a few 6-plexes, it's primarily focused on neurology and the bead technology, the two instruments on the left. The HDX, which we just recently launched, has got an incredible tsunami wave of interest. So we're very excited about the HDX replacing the HD1. And that has got a lot of excitement in our data on the test beds that we have out there and the beta sites that we have is just night and day from the HD1. The SRX is more of a benchtop that just does reading, doesn't do sample prep. And now the SPX, which really, once again, doesn't do sample prep, just does primarily reading, but really it's pretty incredible. It has very few moving parts and it's really a highly reliable technology. And then we have assay kits that we're validating across both the plate and the bead, and we're trying to use what we term coreplex to further validate that we're getting the same kind of results from the plate and the bead and same levels of sensitivity. And we really continue the Accelerator services, which have been growing incredibly rapidly, and we have so many new studies that are underway. We just want to make sure that if there's anything that you would like on a custom assay basis or studies you want us to run for you, that laboratory now does have CLIA, and we're evolving it into laboratory developed test capability. We always try to understand the learnings from our customer base as we continue to advance our roadmap. And we've been learning that sensitivity is in fact vital. And today, this yellow top the second column under SPX, it's yellow versus the Simoa bead-based. Today, we're actually turning that green because we've been able to achieve the same sensitivity levels. And we don't have automation yet for it like we do the HDX for the bead-based. And our menu is not quite as expansive yet, but we're working on menu in both categories. And we continue to feel like the next generation, when we go another 100x on sensitivity, we're already evolving the two platforms we have and feel like there's going to be, the ability to get to 10 to 20x, even with the current platforms that are being bought beyond where we are today. So I wanted to show this because when we started powering Precision Health 4.5 years ago, a lot of it was focused on publications, getting peer-reviewed third-party data that gets published and goes through the scrutiny of a journal's board to allow that study to be presented and published. And you can see how rapidly the publications are advancing, and we've actually now got 149 in oncology, significantly up from where we were last year. So we're pretty excited about the cytokine, understanding these immunotherapies and the possibilities there. And you can see how many markers as well that have been run. Many of these are homebrew. Probably 2/3 of what you see in that second category, markers, are being done via our homebrew capability, which you're going to hear from Mark today. We both have singleplex homebrew on the SPX, but we're also evolving into multiplex. And then in the Accelerator, you can see how rapidly that's advancing. The fifth bar on the right is actually just the first half of this year. You can see we almost have gotten to where we were last year on a full year basis in the first half year. But most importantly is that red line up above shows you how many phase 1, 2, 3 drug trials we're running for pharma biotech, and that's becoming a big area of our focus. Then the number of instruments that were installed now is up to 340 instruments, and you can see that most of those are in neurology, but we're starting to advance in oncology. And you can also see that the growth of our instrument sales has really picked up in the last 4 quarters. And then on the right-hand side, we're really proud to say that one of the biggest features is the pull-through of people actually using the instruments and getting data and getting results. And so the consumables, which are our kits that we sell, have been growing almost 100% a quarter for the last 3 or 4 quarters, which is like off the charts. And that's a key testament to us that the technology is really bringing value when we see people actually using it in that way. So, hey, just to kind of summarize what we've talked about in the past, we're honed in helping pharma companies lower toxicity by getting to disease cohorts earlier, long before their symptoms. And by doing so, many of these trials can be done with lower dose. And the lower the dose, the better the toxicity. And then in the efficacy world, we know that particularly in some of the late stage diseases like cancer and Alzheimer's, it's very difficult to get drugs that actually work when it's detected that late stage. And so we're trying to accelerate cohorts of earlier disease when we think the disease has got a better chance of being dealt with. And pharma biotech's really picked up on this. They've actually done their own analysis that when they're using biomarkers to run trials, if they get a phase 1 approval and they move on to phase 2, there's a 300% increase in the probability they're going to get a phase 3 approval if they use biomarkers. And you can see the 5 areas down below where we've really honed in. And in the area of cancer, going from an 8.4% probability of the phase 3 being approved to up to 25%, we think is a pretty big breakthrough. That's led then to the tsunami wave of CROs using our technology, whether it be Quest or LabCorp or rules-based medicine or Frontage. Everywhere you look now, we've got CROs running major trials. This is just one single CRO has now run 650 clinical trials on the Simoa, but overall it's well over 800, so we're pretty excited. These numbers will be over 1,000 once we get it updated. The FDA themselves, Scott Gottlieb, who's no longer the commissioner, but he will be hopefully coming to our Powering Precision Health in Barcelona. I'm going to be presenting with him next month out in Napa. I'm on Larync is doing a pretty big presentation with all the CEOs from pharma biotech, and so Scott Gottlieb and I both will be presenting some of the advances, but he actually created guidance for biomarkers to be used in the drug development process for phase 1, 2, 3 trials. And so he's really advancing and has advanced the FDA, which the current FDA commissioner has followed through with that. So this next slide just shows all the companies now that are using our technology, either from a services and Accelerator or from buying our instruments, and that continues to grow very rapidly. And this next slide now, I want to move briefly to, there's been a lot of questions about our company relative to NFL. Neurofilament light actually represents 20% of our sales now, and it wasn't even a biomarker in our arsenal 3 years ago. It was a homebrew. And now there's like a big onslaught of trials and studies, and ECTRIMS is coming up over in Stockholm, I believe, in a week or two. And you can see just the ramp that's occurred in a number of publications that were presented. I would bet that it's going to be over 50 this year. And there's 46 active trials now using NFL as either a primary endpoint or a secondary surrogate endpoint, which is further evidencing the advances. So what we did was, based on all that advance, we went ahead and merged with Uman, because they make the antibody pairs that we tried for 3 years to emulate, and we couldn't. And 100% of all the publications in blood are based on their antibody pair, Uman's, and our Simoa. So 172 peer-reviewed publications on NFL, 100% of them are them. And then even in the cerebral spinal fluid plus blood, you can see that a majority are based on the Uman. And on the right side, those were the results we had on all the trials we ran. And we even went to the top 3, Siemens, Abbott, and Roche, to try to source what we would consider to be highly specific NFL antibody pairs, and we couldn't find it anywhere. So we went ahead and acquired them, and we're keeping our focus on, obviously MS, but we're also evolving TBI, where there is a primary endpoint right now with NFL, as well as Alzheimer's. You might have saw the study that some neurologists recently saw Alzheimer's 16 years before dementia, based on a familial trial that got a lot of fanfare. So we feel like NFL is really showing neuronal health. It's kind of like the cholesterol of the brain, or the blood pressure of the brain, to kind of understand across all disease categories if your neurons are being damaged. This is a very good way to measure it. And we're doing a major normalcy study right now, 20,000 different population people, basically from ages 7 to 70, to see what that line is, because we know as you get older, neurons die. So we want to see what the kind of normal curve is, and anyone above that curve, we think in the future is suggesting some levels of issue. And this slide just shows you that we've got a lot of excitement today in our business around analytical validity of many of these biomarkers, but we're trying to move them to the right and get the clinical validity, the FDA sanctioning them, and we're running trials to further support that. By the way, we just moved into, I'm sitting here today, it's 92,000 square foot new facility. It's kind of like the center of biomarker disruption and innovation. So we're pretty advanced now, having everybody in the same location, and almost every day there's customers here that are working with us and partnering with us. So we're pretty excited about how that advance is occurring, and we'd love to see you out here at any moment. And again, if there's questions at the end, I will stick around and look forward to continuing to interact with you around the SPX. Mark?
Well, thanks, Kevin. I'm really excited to introduce everyone to Quanterix next generation technology, this SPX planar technology. And before I describe exactly what that is, I thought it'd be a good idea to talk a little bit about what you need in a biomarker technology or a biomarker platform. And these are pretty obvious things, but I think it's helpful to go through before we kind of dive in and show how the platform works. You're really looking for sensitivity, and some people, they maybe don't think sensitivity is so important because they're looking at a disease sample where there's lots of a biomarker in that disease sample, but the ability to measure a baseline and measure change off a baseline can really change the whole game, as Kevin alluded to in this first slide. So we want sensitivity, we need good reproducibility, CVs below 10%, broad dynamic range, because you're going to want to detect normals at very low levels and then disease samples at higher levels in the same sample. So we're looking for dynamic ranges of 4 to 5 logs, small sample volumes in the low tens of microliters or less if possible. You want it high throughput to do many samples, multiplexing, particularly in the areas of cancer and immunology and inflammation, where it's specifically in neurology, there's a more limited set of biomarkers where some multiplexing is probably very relevant, but high multiplexing is not as relevant at the moment in immunology, oncology, inflammation. People are still doing a lot of studies where they're trying to assess a large number of biomarkers and the shifts in those biomarkers through various different diseases, so multiplexing is more important. You do need the technology to be able to generate new assays as new biomarkers are identified pretty much on a daily basis. You want it to be easy to use. And then ultimately, if that biomarker that you've uncovered in some research effort is going to be used clinical, you need the technology to have a path towards validation for clinical trials. So the SPX, the Simoa Planar platform, our newest platform, meets that criteria. And I'd just like to outline on this slide the components of the SPX imaging system. They include assay kits on the left-hand side here, and we actually provide plates with spotted assays on them and the reagents to finish those assays, homebrew kits, as we mentioned, and the ability for us to create custom kits for you. The platform itself is a relatively simple and straightforward imager, simple, straightforward, robust, no moving parts, and generates excellent results. And then we've also paired that with some automation devices so that you can semi-automate the SPX workflow to provide more consistency and more ease of use, and then put a lot of time and effort into basically what I think of as a customer-centric software package that really works the way customers work when they're analyzing biomarkers, easy to understand, easy to flow through, and will collect all the imaging data from the assays and convert that into concentration values that you can use to assess the actual biology. We do have a range of assays available on the technology today. So we have about 60 standard kits, and they're individual singleplex kits, anything from a standard cytokine like IL-2 or IP-10 through a set of key cytokine and chemokine panels that are designed specifically to get at various specific biological problems. I just point out quickly our flagship product that Kevin mentioned, this CorePlex cytokine panel, is a multiplex 10-plex that's able to detect all 10 cytokines, chemokines at baseline levels in a sample with high sensitivity. When we're talking about high sensitivity, we mean in the low picogram, femtogram per mil range. So we have a set of 60 panels and singleplex markers. We have over 150 other assays on the platform that are also high sensitivity, but they're not available as standard kits. We can make those available at varying levels of validation for individual customer needs, anything from just a research kit through a phase 2 clinical trial validated kit for a customer. And obviously, as we mentioned before, homebrew kits at plates and reagents. So let's get into a little bit about how the technology works. It's another, playing off of the success we've had with our Simoa bead-based platform. It's a simple technology, and some simple things have been done to generate very high sensitivity. So basically what we do is we spot capture antibodies in the bottom of a well, in each well of a 96 well plate. So it's an array of capture antibodies in the bottom of each well. That's very simple, and I'll talk about why we get sensitivity with that in a moment. But once those capture antibodies are on the plate, and we provide the plate as a standard kit, then you would add your sample to the well, come in with a detection antibody, and that detection antibody is labeled with biotin. Then we come in with a biotin HRP, horseradish peroxidase conjugate, as a way to generate the signal. And then finally add a chemiluminescent substrate with a HRP, activates the substrate. We generate light wherever there's a sandwich immunoassay. And again, the beauty here is it's tried and true sandwich immunoassay technology, just as you'd been using, or anybody's been using, for many years. But there's a set of ways that we do this that generate the increased sensitivity. So then we put the plate, the 96 well plate, in the SPX platform. A CCD camera images the signal from each sandwich immunoassay from the bottom of the plate without the effects of the liquid from the top. So from the bottom gives us a much easier and more consistent imaging platform. We take those images, process those images with calibration curves and standards and samples to generate actually concentration curves and generate the concentration of an unknown sample in a typical experiment. The whole process, again, can be semi-automated. It's really easy and simple to use, no real magic. You simply pipette your reagents into the plate. You put your plate on a standard shaker. We have a particular shaker that's optimized for the SPX platform, then onto a washer to do the washing steps. That's repeated as necessary depending upon the particular protocol. Takes about 3 to 4 hours. I do want to point out that it uses the same shaker and washer as you would use for the SRX platform as well. Once the immunoassay sandwiches have been formed and washed and substrate is added, we put it in the machine into the SPX instrument, and it's imaged for between 2 and 10 minutes, depending upon the particular samples, to generate the data. So you say, well, that's a standard immunoassay. You showed how this works, but where do you get the sensitivity from? And that's really shown on this next slide. The sensitivity is way beyond what a standard immunoassay can deliver, and that's because of a few different things. One of the key things is the way we make the capture antibody spots on the plate. We use a special, unique proprietary spotter to spot the capture antibodies in a particular pattern in the bottom of each 96-well plate, in this particular circular pattern you see here on the slide. Using our technology, we're able to spot capture antibodies at a very high density. So we get very high density, very precise spotting in each one of those circles. We've optimized the surface chemistry. We've optimized exactly where on the bottom of that plate to spot each spot. And then we've worked hard to develop some uniform coding and blocking buffers that make just those spots active to capture analyte in the sample. The unique design of that circle, of that circular approach in the bottom of each microwell, is another factor that drives the sensitivity. Exactly where those spots sit, as you can see from the right-hand side of that, is in an optimal region in the bottom of a plate to get consistent amounts of volume and optimized kind of vortex mixing. So we drive because of where that capture antibody sits, we drive the reaction binding kinetics very efficiently. And that plays a huge role in driving this increased sensitivity beyond a standard ELISA. And we avoid problems when people tried to do this in the past. You know, if there's a spot in the middle of the well, there's a different amount of volume. And then when you go to image, you've got some diffraction issues. So we've taken away all those problems, and we get very efficient, very high sensitivity detection and spotting. Again, simple format. Once that plate is set up, you put it in the SPX instrument itself. That instrument has kind of like no moving parts, so it's very robust, works all the time. You can simultaneously image a whole plate. We use a high sensitivity scientific grade CCD. There's no excitation light. And that's another place where we get this higher sensitivity because we're measuring signal from the sandwich immunoassay on a dark background, a completely dark background. So there's no need to filter out anything. Each photon that gets generated, we can detect. Again, that drives the sensitivity and makes the instrument really robust. So the workflow for that is relatively straightforward. You put a plate into the instrument. The instrument does a short exposure to evaluate signals from the calibration wells, then does another short exposure to look for analytes that are in high concentration, then does a longer exposure for analytes that would be to capture every photon, to look at analytes that are in very low concentration. And then, again, as I suggested earlier, we worked hard on all these algorithms to generate a composite virtual image from each of these images that is able to quantitate both low sensitivity, low samples and high samples at the same time. The total read times between 2.5 to 10 minutes, depending upon the particular experiment that we're doing. And then to kind of give you some sense that, yeah, this technology really is sensitive and it really does compare well with the bead-based technology. Here's an example of the detection of IL-6, a very common cytokine, using the HD1 platform, which you know and love, generates very high sensitivity data. And when you compare that to the same assay on the SPX platform, you see we get excellent correlation of data, an R-squared of 0.98. Very similar data in both platforms. Essentially, the data is the same. The sensitivity levels, the LODs are roughly the same. The functional LODs are nice to talk about in terms of sensitivity, but what you really need in an experiment is the LLOQs and the limit of what you can really quantitate. And the limit of what you can quantitate on the SPX is about 30 femtograms per mil in this particular experiment for IL-6, and it's a little bit actually higher for HD1. So excellent results with the SPX platform, similar sensitivity, and we've proven that time and time again. The other point I'd like to make is that it's not just that ultra sensitivity that's important. Well, it is important for this reason, and the reason is detecting the particular analyte in normals, in 100% of normals. So you see in this data set that the percent of samples that are above LOD and above LLOQ is 100% for both technologies. So we like to kind of say around here is to use our technology to not be a BLOQ or not be a bloke, below levels of quantitation. So now to show you some data, some real world data of the technology, I mentioned previously that core cytokine 10-plex, and we're very excited. It's kind of our flagship assay. We worked very hard for a year and a half or so to optimize this assay to make it so that we could detect all 10 of these cytokines, interferon gamma, IL-1 beta, IL-4, IL-5, IL-6, IL-8, 10, IL-12, P70, IL-22, and TNF-alpha in one sample at very high sensitivity and detect 100% of the samples. So we have that kit available for sale today on the SPX today. It's a great product. We worked in collaboration with some well-known oncology KOLs to develop the platform. This is some data that was generated independent of us using this assay against some gastric cancer samples and some colorectal cancer samples. Again, we were able to detect all these cytokines in all the normals. So we had a baseline to work off of, and you can kind of look at the pattern. There's different patterns for each particular case. But being able to see all the normals now gives us the ability to evaluate what is going on with each one of these cytokines. Really gives you a handle on the biology of what's occurring in that particular cancer sample. So I think that's really pretty exciting. I wanted to go through just a few more specific application examples to give you a sense of the power of the technology of being able to detect these cytokines and chemokines and other biomarkers I actually chose some immuno-oncology examples to really show you where it has an effect on somebody's study. So this is an example of the detection of about 15 biomarkers in a phase 2 clinical study that was done in the UK to look at biliary tract cancer after treatment with cediranab. And what this particular paper suggested, this Backen et al. Paper, was that treating with this cediranab inhibitor, which is a VEGFR2 inhibitor, they could monitor the inhibition of VEGFR2 as you would expect. They profiled a set of 15 different angiogenesis and inflammatory biomarkers and could look at those patterns over time and correlate it to the result of treatment with cediranab. So, the clinical value in this paper, the importance of being able to do this is now there's a tool to easily measure circulating biomarkers to provide an alternative to, having to keep taking biopsies and looking in a particular sample for a biomarker over time, right? So it makes it a usable tool to follow and track an important clinical study. Another example is shown on this slide, a paper by Secord et al. In this study, it was a phase 2 trial with nintedanib and looking at ovarian cancer, 27-patient phase 2 study, looked at a wide set of biomarkers, again, using this technology and measuring these biomarkers at baseline. Now, if you try to use a lot of other technologies, when you go to do the experiment, you're going to get a whole bunch of these BLOQs, right? You're not going to see the normal. So you can see a particular biomarker when it is highly elevated, but how about subtle moves? That's really going to tell us where the biology is. So in this particular experiment, in this particular paper, these guys were able to show that CD73, IL-6, and VEGF-D were prognostic for the effects of nintedanib in this particular experiment. It's correlated with the disease slowing. So again, just a nice example of monitoring the biology with with high sensitivity biomarkers. The next example, everyone is pretty excited about CAR-T technology. It's a paper by Young et al. In Cytokine Research last year, where they were using CAR-T therapy. And one of the challenges with CAR-T therapy is sometimes it works really great and sometimes it doesn't work so good, and nobody's really sure. And we need biological handles to monitor what's happening with CAR-T therapy. So this group was able to profile this set of cytokines using the SPX platform: interferon gamma, IL-2, TNF-alpha, IL-10, IL-4, IL-6, and again, correlate that back with the ability of CAR-T cells to activate an immune response and actually get at and reduce this B-cell lymphoma. So again, being able to measure this at normal levels and profile those low levels as they change over time provides us with a tool to really understand what is going on with CAR-T therapies. Last example I'll give you is another example to look at how you could maybe even use biomarker, ultrasensitive biomarker technologies to identify new targets. So this paper by DeMarchi recently in Oncogene, they had a hypothesis that the P2X7 receptor influenced the tumor microenvironment. We all know that for tumors to grow successfully, they invade the tumor microenvironment. There's a whole component of activation and degradation of the tumor microenvironment. So these researchers used, again, the SPX technology, measuring IL-1 beta, TNF-alpha, and interferon gamma to monitor the immune response and correlate that with the degradation of the tumor microenvironment. And they were able to show through this set of experiments that P2X7 really did play a strong role in tumor infiltration. So using these cytokines, they essentially identified a new target. Very, very powerful technology. Okay, so those are the key application areas I wanted to go through with you, but what about if you have a new biomarker and you don't want to just profile these standard, well, maybe you do in some other setting, these standard cytokines and chemokines, but you've got some new hot, particularly there's everyday new oncology biomarkers that are being discovered. You want to build an SPX assay for it, and you want it to be ultra sensitive, as we've shown for the assays that we have today. We do have the homebrew kit. As we mentioned before, it works relatively straightforwardly. We provide homebrew assay plates, which have an anchor peptide. You take your antibody to the biomarker, your new biomarker of interest, and you tag it. And that tag then will be specific to the anchor, and you effectively use the same sandwich immunoassay format and generate a single-plex homebrew assay. Technology works really well. It works almost as well as spotting the initial capture antibody directly on the plate itself. I say almost. It's pretty darn close. This is an example of IL-6 done by a homebrew and by direct. The data lines up essentially on top of one another. I have seen examples of even the homebrew assay being slightly better than the direct assay, maybe because of some steric interactions in a particular setup. But again, technology works really well, really sensitive. We've done it for a host of capture antibodies, including mouse, rabbit, rat, goat, so it seems to work well kind of across the board. So definitely, if you've got a new analyte that you'd like to measure and track, the homebrew approach to SPX will work well for you. It's relatively simple, straightforward, easy to use, and as I suggested, really positive results. We're really excited about the development of multiplex homebrew SPX technology. So I show that on the next slide here. The same concept that I showed in the first slide. It's just that you use a different tag for each different capture antibody. So this is an example of a multiplex homebrew 5-plex, I guess, is how we would say it. The concept is pretty simple, straightforward, works as I suggest. The single-plex works because actually can multiplex by spotting locale. And we've just started developing and optimizing that technology ourselves here at Quanterix. We've got some excellent feasibility data, which I'm showing here on the next slide, measuring IL-5, IL-6, IL-10, IL-22, and TNF-alpha, and comparing a direct print format to this multiplexed a homebrew format, and you can see again the correlations really good. 0.99, 0.92, 0.97, really nice. So we're really excited about this multiplex technology because as we've been working with customers, we have these panels, and the panels are excellent products, but people will frequently say, yeah, I like that panel, I got this set of work done on that panel, but now I have these 3 others I'd like to try. And now we have a format that you could mix and match and create your own panels that will be very specific to your particular application. So that's a product that's in development. We think we'll have kind of a kit-based format for that kind of around the first of the year timeframe. So that's the SPX technology in a nutshell. We're really excited about the technology in terms of multiplexing, in terms of its advantages in cost and ease of use. I've shown that it can detect in the femtogram to picogram per mil range, high sensitivity. I showed lots of data on detecting baselines. All the data typically is below 10% CVs, has wide dynamic range. Use about 12.5 microliters per sample. Again, I showed the throughput and the multiplexing, homebrew, and we're working as well to create a path for this technology. We use the technology ourselves in work we do for other clients in our Accelerator lab that's preparing for clinical trials. So that's in a nutshell how the technology works and some data examples. And frankly, what we'd like to do now is take some specific questions. And we've got both Kevin and myself and some technical experts here as well that could answer some of the questions that you guys have about the technology or its applications.
So Mark, I've been taking a little bit of a look at these questions while you were speaking, so maybe I could start to comment on a few of them. Is testing with this technology offered by CROs? And today, there's one CRO that does, PacBio.
That's correct, yep.
But it would seem like, certainly in our own operation, which is another question, we absolutely can run CRO services with this technology. I would fully expect that the other CROs will be, very short to have this technology as well. There's a question, is it possible to run single cell assays with sorted cells? And so Mark, I'll start and maybe follow up, but we don't have any what I'll call single cell sample prep on the front end of this, but if you guys can get into a single cell, we lyse it open, certainly that would be something that could be achieved, but what's your view, Mark?
Yeah, my view here is that there's enough sensitivity in the platform that if you have a way to isolate single cells, there's enough sensitivity that you should be able to detect individual proteins within those cells. We've demonstrated that on the bead-based technology, and we have actually some publications in that regard. So I expect that it would work with the SPX. I don't personally know of an exact example of it yet, but I expect it would be feasible.
So one question, I love this one. It says, is SPX sensitive as the HDX or not? Question mark. At the beginning, it did not have a green light on sensitivity. That was my chart. And what I'd said there was that for the last year since acquiring Aushon, we've applied a lot of the sensitivity technologies to Aushon's basic multiplexing platform. And that's the breakthrough. It's the 1 + 1 = 5 relative to achieving this level of sensitivity with our multiplexing platform. And the answer is yes. But the answer hadn't been yes until probably in the last 3 months. And that's been a key to our advancing. But it leads me, Mark, to want to ask a follow-on question. And David Duffy's in the room here too, our Chief Technology Officer. And I get this a lot, and I'm not sure how to best answer it, but the industry has traditionally seen a trade-off between multiplexing and sensitivity, meaning the more plexes that you run, the more you give up in sensitivity is somewhat a traditional view. And what we've been trying to do is shift that curve on sensitivity and multiplexing. But I guess what my question is, how much sensitivity do we give up if you want to, let's say, the 10-plex panel mark that you've been showing, if you were going to run that as a single plex versus a 10-plex, my understanding is that it's no different than the bead technology to date, meaning that the amount you give up is relatively consistent, but you don't really have the ability to run a 10-plex today with the bead technology. So
Correct.
Could you or David comment a little bit about this trade-off of multiplexing and sensitivity?
Yeah, I'll take that one, Kevin. So you're right, there is a trade-off, and I think it's like in two areas. One is sensitivity detection, ability to detect the molecules, and with the beads, we would run into the problem of you split the beads among more analytes, and then that means that you are sacrificing some sensitivity because you don't get as many beads read. With the planar platform, you don't have that issue because they're all just spotted at the bottom of the well, and they're all experiencing the same kinetics. So kind of it's easier to scale up multiplexing in the planar format, which is why we're focusing in that area. The second area applies to both, and that's buffer optimization and reducing non-specific interactions and cross-reactivities. And that'll apply to both platforms. And I think the really nice thing about the CorePlex is there's an awful lot of effort went into the blockers and the buffer composition so that you didn't compromise anything in sensitivity. And that's not easy to achieve, and it was our assay development group. Now, if you go to 30, it becomes even harder, but you know, in the 10 range, we've demonstrated that we can do it without compromise. But you're right, it's very hard. Yeah. So
David, that was helpful and pretty insightful. One thing I've been hearing is like some of the competitive platforms are only running single plex, but then they can run like a 10-plex by actually splitting the sample 10 times and running each one of them down their own channel.
Yep.
But by doing so, it does seem to me that that's where you really end up giving up a lot on sensitivity, or you have to have a hell of a lot more sample in order to get the 10-plex answer. And it felt to me like this is a compromise that we tried to innovate here is how can you get a 10-plex answer from a very small sample without having to actually split it 10 times or even split it, 2 or 3 times. You want to comment on that? Because I think it's a pretty important advantage that we tried to get to with this compromise.
Yeah, I mean, different people have different ways of going at the problem of the compromise, and the example you give there, you split things up so you reduce cross-reactivity between reagents, but then you do give up nothing is free, right? And so you end up giving in sensitivity because you've split your sample, and how scalable is that? How easy is and then sample volume? You're needing more sample volume. And the approach we've taken with the planar platform is, well, let's really try to get it to work in one well, where the capture reagent is experiencing all of those molecules in the sample, which isn't the case when you split it up. And I think it's really through the chemistry and through the assay optimization that has allowed us to get there with the 10-plex.
Excellent. And I think that trade-off in trying to innovate came directly from many of our customers. They want to be able to get an answer with the least amount of sample, because many times in mice particularly, you got very small samples, but they want that multiplex, but they don't want to give up sensitivity, and they don't want to give up any of that by diluting the sample to be able to split it. And so we don't have to give any of that up the way we've engineered it. What's the size, Mark, of the sample needed for the SPX compared to the HD1? Because that might also, maybe the answer here is better articulated once the answer to that is understood.
Yeah, so the typical sample volume for the SPX is about 12.5 microliters, and on the HD1, it's more like 32 microliters or so.
So to me, that's where this breakthrough starts to occur, because if you can get 10 answers from that amount of sample size,
Right.
and keep the sensitivity where it is, then you really have magnified the sensitivity because you're not needing to use any dilution to run the sample. So anyway, I just think that there's a breakthrough in there that may not be as obvious for people to see, but that sample size being so small to me is a pretty important breakthrough for that level of multiplexing,
Right.
and maintaining the sensitivity. Mark, another question, and you might have them over there too, but there was a question around, something around a phosphate.
Is there a phosphate protein assay available? So I believe what someone's asking there is, can you detect phosphoforms of proteins? And the answer is, well, yes, you can detect those. If you have an antibody that's specific for a phospho group on a particular peptide or protein, you can absolutely develop an SPX homebrew assay for that. We don't have a specific phospho assay on the menu at the moment, I don't believe, but I know some customers that have done that with the technology. So definitely possible, but you'd need a good specific capture antibody.
And Mark, our Accelerator lab can build custom assays, either singleplex or multiplex for anyone very rapidly if they want to kind of get at that answer quickly. And another question came through was, people want demos that are on this call here and wondering, like, how can we be most efficient in getting demos quickly?
Yep. So we can definitely do demos. It's the easiest way is to come to this beautiful new facility that Kevin mentioned to have a demo. We have labs up and running here. We can do rapid demos pretty quickly. If that's not possible, we have several other sites that have instruments. We could bring clients to those sites, and then in the last case scenario, if you have a really genuine interest and like to see the technology, the instrument's pretty portable. We can come in and do a demo directly in your lab. And the best way to do that is just to send an email to sales@quanterix.com, and we'll coordinate that with you rapidly.
And so, Mark, I found these next two questions to be pretty interesting. One is, how would you compare this to microarray ELISA or Lispot? And it would seem to me that sensitivity might be part of that answer, but I'd like to get your views on that. And then also just the pricing information on the instrument and the plate. I think they might be interested to learn how cost efficient this technology is.
Yeah, so with regard to the answer to your first question about comparing to Lispot technology, again, very similar technology, but we've engineered over time, over a number of years, this very precise high-density spotting technology based on our kind of proprietary spotter, and then all the buffer work and then the positioning. It drives the sensitivity to a level that's a little bit above an Lispot kind of sensitivity. So I think people do use Lispot occasionally to detect proteins from single cells, so I'm quite confident that our technology should be able to do that as well.
So Mark, why not use Lispot or microarray all the time then? Like, it sounds like you're saying that the sensitivity of Lispot and microarray can start to approach what we're doing, but I would thought there's big workflow advantages for throughput in the approaches. And curious, and Jeremy's also here too, as the lead product manager around the technology. I know you've done some studying in this area, but Jeremy, you have any views on that?
Well, most of the assays have been developed and optimized for detection in serum and plasma or other sample types rather than the Lispot, which is measuring protein secreted from the cell on the plate. So when we saw the examples in Mark's slides from some publications looking at serum and plasma biomarkers and the relevance there, and that's really only enabled with this technology format. The question about how it compares to some other microarray ELISA technologies relies on some of the attributes of how the spots are printed, how the chemiluminescent reaction is generated, the signals generated. There's a paper that was recently accepted for publication in Journal of Immunological Methods, which goes into fairly significant detail describing the technology. So if anyone's interested, we can point you to the reference for that paper, and you can learn more about the fundamentals of the technology.
And Jeremy, to that point, we've got, I think, this is correct. If it isn't, I hope that it'll be correct very soon, but all 550 of our third-party peer-reviewed publications are actually somehow indexed on our website, correct?
They are, yep.
So there's different ways you can sort to get at different publications. I had some investors the other day asking me about autoimmune and immunology. Do you have any publications in that landscape? And I don't know if anyone's gotten to you, Mark, but we do probably want to index for them all the ones that deal with that, because it's, I think, a key to our approach to evolve our company is to rely on our partners out there that have done and invested in all these third-party peer-reviewed publications. We think it's the best way for many of our scientific customers to link in. And that brings me back to Powering Precision Health, which is an independent nonprofit that is independent of Quanterix, but Quanterix is a sponsor, as well as many other companies sponsor. I think the last one we had Pfizer and Biogen and Quest and others also sponsor, as well as some of the banks. It's key because that's where we inspire the whole concept of publications. And we have many of those innovators there personally for 2 days that present across oncology and neurology tracks. We also have patient advocacy groups there from oncology and from neurology. We also have investors that attend that can look at these different technologies, talk to all the customers, and then we have all of our prospects, people that want to learn more can come, and it's a very efficient way to kind of get deep into the publications and understand them from a lot of direct discussion. So anyway,
yep.
That is correct around the indexing, correct?
Yep, absolutely.
Perfect.
Now, the question I want to make sure we answer before we run out of time is somebody's asking about pricing info from the instrument and the plate. So the instrument itself is $75,000. Robust, solid instrument, no moving parts, will be reproducible and robust for anyone. And then depending upon the particular assay, a plate's worth of assays starts at about $500, and some of the more expensive ones, the multiplex 10-plex would be about $1,500. So that's the kind of price range. But I'd also like to point out as well that the reagents in this format, when we produce the reagent kits, they're very stable. So they're stable for beyond years. So that creates a lot more flexibility in what you're trying to do. Some particular biomarker reagent kits expire quickly, and these don't. So that's an advantage as well. Okay, we're looking for a few more questions here that we might be able to answer. So it looks like a couple different people are interested in how you might be able to use the technology to detect a truncated protein missing the epitope for detection. So I guess that's a tough question. If you don't have either a capture or detect antibody that's specific for something that's left on that protein, you're not going to be able to see it in the technology. So you would just have to, but that is one thing that our Accelerator group works on extensively. When people come to us with some new analyte, and we've done this quite extensively for tau, because the tau biomarker as a neurodegeneration biomarker is processed extensively, and it gets to both questions that were asked. Certain people want to look at particular phosphoforms. Certain people want to look at truncated forms, mid-domain forms. And so our Accelerator team works very hard to make sure they have that particular antibody reagents to detect many different isoforms of that protein. And you can make an assay that works for a particular isoform very effectively. But if you're trying to detect a particular fragment that's cleaved off and absolutely not there in the sample, you're out of luck.
So Mark, one question I wanted to throw in here. I think 2/3 of the demographics of who's attending this are oncology-based. There's a big move toward liquid biopsies, and we've got some liquid biopsy companies that have now bought into our SRX technology because it also can play a role in doing DNA and RNA. As well as proteins. Now, I was just curious, do you ever see the possibility of our SPX being able to do similar kinds of exquisite sensitivity without PCR, or get to the same level of sensitivity as, say, digital PCR, but doing it with a native DNA, RNA sample. Do you ever see the SPX having any of that versatility?
Well, I would say that absolutely it's feasible, because the printing technology that is used to make the plates for the SPX protein arrays can be used very effectively to make nucleic acid arrays. So you could leverage that capability and that sensitivity. I think there would need to be some development work to make the protocols actually work, and we haven't actually done that ourselves. So,
but from the concept of a homebrew, which is where I think a lot of it's materialized on the SRX, is that a place that maybe there's a possibility of utilizing the instrument?
It could be, and if people are interested in that, maybe we'd love to talk to them in more detail.
Yep.
And see if there's a possibility to collaborate around that.
Perfect. All right, so it looks like that is most of the questions, and our inside sales team can follow up on other questions that weren't answered here, or if you have additional questions. Really want to thank you for attending today, and we're really excited about this SPX technology, as well as the HDX, which we just recently launched. So thanks again, and we'll see you soon.
Bye, everyone.